Colleagues, friends, and family of Peter van Dijk. My name is Jiří van Thune. I'm the CEO of KeyGene, and I herewith would like to welcome you all here in Wageningen at KeyGene, or online by the Teams connections on this Mendel Day 2023. I want to also welcome you on behalf of the Mendelianum of the Moravian Museum, located in Brno, Czech Republic, represented here today by Dr. Jiří Mitáček and Dr. Jiří Mitáček. We celebrate the life and work of two important geneticists. First, the life and work of Johann Gregor Mendel, the founding father of the field of modern genetics, which was and is tremendously important for our plant breeding and plant sciences sector. And second, the life and work of Peter van Dijk, geneticist and distinguished scientist here at KeyGene, and laureate who received the Mendel Memorial Medal Award. But before we enter in the Mendel Memorial Medal Award ceremony, the long sentence, we have a kind and warm up by two presentations. First, we will have a presentation which is more from a historical perspective on the life of Gregor Mendel. And second, we will have a presentation on the latest and striking developments in human genetics. Subsequently, we will have the Mendel Memorial Medal ceremony, followed by a presentation by the laureate Peter van Dijk himself. The program will end at 5 o'clock with drinks and bites. I hope, and I will also check, that at the end of the program, I will see a smile on the face of each of you. And I also trust that you are proud of the life and the works of the two important geneticists, Mendel and van Dijk, who we are going to memorize and celebrate today. And with that, I would like to present the first speaker, Professor Dr. Daniel Fairbanks. He previously was Dean of the College of Science and Interim Dean of the College of Technology and Computing at the Utah Valley University. Prior to joining the UVU, Professor Fairbanks worked at the University of Massachusetts, Amherst, Brigham Young University. He is a geneticist specializing in evolutionary genetics, the history of genetics, plant genetics and breeding, and also international scientific development. He is author and co-author of many scientific publications and scholar books, and he has more than 60 scientific papers on his name on the same type of topics I just mentioned. He's recognized as one of the foremost scholars on Gregor Mendel and is author of a recent biography of Mendel. The Fairbanks is an accomplished artist, on the other hand, and he is also doing sculptures, paintings, and drawings in museum, public, and private collections. His works are there in the United States, in Latin America, and in Europe. And he has received numerous awards. I'm looking forward to your presentation, which is entitled you have changed the title. Okay. A brief review of Mendel's life, influence of Carl von Gärtner, Mendel. I'm looking forward.
Thank you very much for that introduction, and I'm very honored to speak here, especially in honor of my friend Peter van Dijk. I'd also like to acknowledge two students, Hannah Thomas and Madeline Tonioli, who have contributed to this research. They are students at my university. Is this the right tool for advancing the slides? There we go. Needs to be turned on. All right. Also, much of this will be based on a new biography of Mendel that I authored this year in honor of the bicentennial. Mendel was born in 1822 And so this book is a new biography to celebrate that bicentennial year. Also, I could not give a presentation like this without acknowledging the assistance of Dr. Anna Matalova, who in 1993 encouraged me to start doing research on Mendel and has assisted ever since. She's now the emeritus head of the Mendelianum at the Moravian Museum. That's a photograph of me with her in 1993, and then the second one was one taken in 2017 when I received the Mendel Memorial Medal. And also, I wish to give special recognition to Dr. Peter van Dijk, whom we honor today as the Mendel Memorial Laureate. He is a distinguished scientist, a premier Mendel scholar, a generous colleague and a dear friend. He and I have collaborated on quite a number of projects and will continue to do so. I also wish to acknowledge what we sometimes informally call the Mendel team, which is a loosely organized group of scientists and historians and people interested in Mendel. And these are photographs from past Mendel days. The wonderful thing about this team is that we all collaborate with one another in helping to promote the new discoveries on the history of Mendel and promoting a better understanding of the accomplishments that he made. So for the first half of my presentation, I'm going to give a brief review of Mendel's life, and we'll follow to some extent the chapters in the book. And many of the images that I've included in here are my own artwork, much of which comes from the book as well. So if we begin with Mendel's birth, he was born in what was then a village named Heinzendorf, is now Hinčice, near the Czech-Polish border in Silesia. This is a painting of his that I made of his birth house, depicting it more or less how it appeared around 1900. We don't quite know what it looked like during his day, and he was the second child in a family of five. Two of his other four siblings were sisters. Two of them died very young, one as an infant, another as a toddler, and then two of his sisters lived to adulthood. We have some only one faded photograph of Veronica, some other fairly poor photographs of Teresia, so I've used a little bit of artistic license to depict them as they might have been. And then, of course, of Gregor Mendel, we have many photographs, and I did the painting here from the most famous of those. He attended school at four schools. The first was the village school, which is almost literally a stone's throw from the home where he was born. And his teachers there recognized how intelligent and precocious he was, recommended his parents that he be sent away to boarding school. He attended a boarding school in Leipzig for 1 year and was listed as top of his class. And then he attended from age 12 to 19 at the Troppau, what's now Opava, gymnasium, and also excelled very well there, even though he had a severe nervous breakdown while he was there and had to retreat home. Nonetheless, he performed well, which qualified him then to enter the Philosophical Institute in Olomouc, where he was there for ages 19 to 21, but interestingly also suffering a severe nervous breakdown, which caused him to retreat home. Yet nonetheless, he still excelled very well in his schooling, and his physics professor there recommended that he become a friar at the St. Thomas Monastery in Brno. And so we'll turn there. From the years 1843 to 1851 was when were his first years at the monastery. This is a painting of it that I made, and in it included the greenhouse, which no longer exists. But also one of the wonderful things about being an artist, if you look at just in front of the greenhouse, was the garden where Mendel did his experiments. Most people think that it's actually closer to the monastery, but it was located on the south of the greenhouse, and I painted in there tall and dwarf pea plants with white and purple flowers to show Mendel's pea plants there. He went through training at the Theological Institute and in preparation for his ordination to the priesthood. And he was then appointed to be a parish priest, but his abbot wrote that he is much less fitted for work as a parish priest, the reason being that he has ceased with an unconquerable timidity when he has to visit a sick bed or see anyone ill or in pain. Interestingly, these nervous breakdowns were coming about once again. Now, one of the fascinating things about this is just this past year, scientists at Masaryk University sequenced Mendel's DNA and got his genome. And in it is a variant that is associated with epilepsy and nervous breakdowns. So this may well indeed have been an inherited condition, just very recently discovered information. Fortunately for science, then Mendel could not continue as a parish priest. As a consequence, Knapp assigned him to be a teacher at the Zoinmo Gymnasium in 1850, and he excelled there as a teacher. He was like a fish in water. He was very much a natural teacher. However, all teachers were required to be certified through a three-part examination. And so this examination consisted of homework, an on-site written examination, and an oral examination at the University of Vienna. Well, he, according to one of his examiners, Christian Doppler, that they found that for the time being, they had to declare him unqualified to teach physics. And Rudolf Neher, who examined him in natural history, determined that he is not yet competent to be a teacher. It is very fortunate for us that he failed this first examination, as Vyacheslav Orel put it. Had he passed his teacher's examination, he would have stayed on at the Zoinmo Gymnasium, and several generations of secondary school pupils would have gained an excellent teacher. Science, on the other hand, would almost certainly have lost one of its leading discoverers. So let's turn our attention now to Mendel in Vienna. He was supposed to spend only a year there, but ended up spending 2, and the timing could not have been better. He studied with some of the foremost scientists in the world at that time. Most of you may recognize the name Christian Doppler, after whom the Doppler effect is named. Mendel's very first courses were with Doppler, and in fact, Doppler died during the time Mendel was at the University of Vienna, so Mendel was one of Doppler's last students. He also studied with Eddingshausen studied combinatorial mathematics, which was very important to his experiments. And in fact, Eddingshausen was a leading authority on combinatorial mathematics. And then importantly, he studied with Franz Unger, who was a botanist, but also teaching evolution in a very Darwinian way before Darwin. The years Mendel was there were 1851 through 53. Darwin's Origin of Species was published in 1859. And so they were unaware of Darwin, and yet if you read Unger's writings, he was very much a Darwinian type evolutionist. In fact, we should probably call him an Ungarian evolutionist, because Unger's published evolution precedes Darwin. I won't read through these, but these were the courses that he took. They were in physics, chemistry, mathematics, botany, zoology, instrumentation, microscopy, and paleontology. So a very rich education that Mendel received at the University of Vienna. So with that, let's turn to his experiments. He returned to the monastery in 1853. And then in 1854, he purchased 34 pea varieties locally and grew them for 2 years to ensure that they were true breeding. And then from those, selected 22 for his famous experiments. He then completed his experiments in the year 1863, prepared them for presentation and publication, and he gave his presentation at the Natural History Society of Brno, and which the Natural History Society was a fairly young society at the time. It had been in place for 4 years, or this was the 4th year of the society when Mendel gave his presentation there, and he was one of the founding members. And so the society held monthly meetings. It's interesting to look at the monthly meeting prior to Mendel's presentations. His were in February and March of 1865. But the month before was Alexander Makowski, who presented on Darwin's theory of organic creation. And the entire presentation was focused on Origin of Species, highlighting passages from that book and a very positive approach to Darwin's book. So Mendel was then the next featured speaker for the next two presentations, for the February and March one, but on February 8th and March 8th, which is why we hold this celebration today on March 8th, is to celebrate that second presentation. And Mendel focused on 7 pairs of characteristics that were easily distinguishable in pea plants, and then hybridized plants of those 2 characteristics together to observe what happened in the progeny. And he initially began, as he put it, with plants were used which differed in only one essential character, which is a good scientific design, meaning that they were that each cross consisted of parents that were the same for 6 of the characters and differed in only one, meaning he had 7 of these monohybrid experiments. And then in the first generation, he observed that all of them had only one of the 2 characteristics, but in the second generation, then a famous 3-to-1 ratio. And when he carried it then to the 3rd generation, which by the way, he called the 2nd generation from the hybrids, he observed then a 1-to-2-to-1 ratio, which begins to show this symmetry of combinatorial mathematics. And by the way, a lot of people don't realize that he carried these through self-fertilization up through the 6th and 7th generations, and then described mathematically the ratios that should occur with repeated self-pollination. And then he did a dihybrid experiment for seed shape and seed color, combining 2 of the characteristics together, and observed a 9-to-3 to 3-to-1 ratio in the 2nd generation. And from that, he then carried it then to what we now call the F3 generation, and observed this 1-to-1 to 2-to-2 to 4-to-2 to 2 to 1-to-1. And you can begin to see it's approaching normal distribution, or what we would sometimes call the bell curve. And then he did his most extensive experiment, was a trihybrid experiment. And again, here is the what we would call the genotypic ratio that comes from it. I will not read through all those numbers, but again, you can see them approaching this this symmetrical normal distribution. Very heavily focused on the common type of combinatorial mathematics. This really was unique for his day. Hybridization of plants, looking at progeny, was not, but this application of combinatorial mathematics was, and he used that then to develop a very extensive theory. These are his words summarizing his experiments. The progeny of hybrids in which several essentially differing characters are united represent the terms of a combination series. Constant characters occurring in different forms of a plant genus can, through repeated artificial fertilization, occur in all possible combinations according to the rules of combination. Notice the heavy combinatorial mathematics approach that he's taken. And now, we typically attribute certain what we might call laws or principles to Mendel, usually the laws of segregation and independent assortment. But what did Mendel perceive as his law? And in my opinion, as well as that of several other scholars, including Dr. Sakharov, who is here, what Mendel perceived as his law was that one germ cell and one pollen cell unite into a single cell that is able to develop into an independent organism through the uptake of matter and the formation of new cells. This development takes place according to a constant law founded in the material nature and arrangement of the elements. This, by the way, was not commonly accepted, that one germ cell and one pollen cell unite to form, or one egg cell and one sperm cell in animals and humans, to form what we now call the zygote. Mendel's results could not be explained any other way than if this happened. By the way, Darwin did not accept the idea of equal inheritance from both parents. So this was truly a major discovery that we pretty much take for granted today. But also important is this last sentence referring to the material nature and arrangement of the elements. And Dr. Sekarak, by the way, has suggested that perhaps material composition or material nature might be a better translation of the of Mendel's view, but nonetheless, he understands that there are chemical substances that are united in fertilization to result in the sorts of patterns that he observed in the progeny. Nonetheless, although some historians of science have argued that Mendel did not articulate the principles of independent assortment and segregation, in my view, they are in fact in his paper. And here is Mendel's description of segregation. The differing elements succeed in emerging from their compulsory association only during the development of the reproductive cells. In the formation of these cells, all existing elements act in a completely and free and uniform arrangement in which only the differing ones reciprocally segregate themselves. In this manner, the production of as many germ cells and pollen cells would be allowed as there are combinations of formative elements. We could hardly state the principle of segregation better in a textbook than Mendel did himself. And then independent assortment appears several times in the paper. This is just one of those passages. The behavior of each pair of differing characters in hybrid union is independent of the other differences between the two original plants, and further, the hybrid produces as many types of germ and pollen cells as there are possible constant combination forms. Now, Mendel and Darwin were contemporaries, and there's been a lot of speculation about what was their relationship, if any. I can answer that question very directly. Mendel knew of Darwin very well and read Darwin's books, including Origin of Species. He had his own copy and made annotations in it. Darwin, as far as we can tell, knew nothing of Mendel, even though he actually owned a couple of books that had very brief references to Mendel, but no evidence that Darwin knew anything of him. This is one of Mendel's annotations in his copy of Origin of Species, which in Darwin's original English reads, the slight degree of variability in the hybrids from the first cross or in the first generation, in contrast with their extreme variability in the succeeding generations, is a curious fact and deserves attention. And those who are familiar with Mendel's experiments would know why he marked this. This is Darwin essentially stating what Mendel discovered. And Mendel, by the way, discovered what that curious fact is. Now, it's sometimes said that Darwin had a copy of Mendel's paper, a reprint of it, but that it was uncut and therefore unread. It turns out the idea of uncut reprints is not correct. There were printer errors in the reprints, and Mendel cut them and made these hand corrections, which are quite unique. They are present in several reprints and therefore indicative that Mendel was the one who cut and marked these. But nonetheless, there's no evidence that Darwin even had a reprint of Mendel's paper, and no evidence that he knew anything of him. Now, Mendel was reluctant to conclude that what he had discovered in peas and beans in his original paper was sufficient to draw any kind of a major theory from it. And so he ultimately said in a letter that a final determination will result only when detailed experiments on the most diverse plant families are available, and he was not content to leave that to others. So instead, he conducted a series of experiments. The most famous of those, which Dr. van Dijk will discuss later today, are the hawkweed or hyraceum experiments. Some people have concluded that Mendel was seeking to make a confirmation with hyraceum. In fact, he intentionally chose plants that he knew would not follow the principles that he had discovered in pea, in hope to come up with a broader theory of inheritance. And yet some people have claimed that these experiments with hawkweed were disappointing. They shattered the hopes he had entertained of finding a confirmation. The worst possible choice, a completely misguided choice of a plant for Mendel to work on, and the results were a mess. They were not a mess. They were actually very detailed, with some extraordinary discoveries that come out of them, and that he ended up publishing later and that are available in some of the letters. But also he didn't work just with hawkweed. These are the other plants that he did hybridization experiments on. And he did in fact confirm the patterns that he observed in the pea plant. This is his statement that for stalks, 4 o'clock and maize, their hybrids behave exactly like those in pisum. And so he did in fact confirm his observations. And in fact, the only disappointment is that Mendel didn't publish much of this additional research that he did. He was elected abbot in 1868. Some historians claim that that brought an end to his hybridization experiments. It didn't. He continued them through 1873. But nonetheless, his work as abbot did in fact distract him from some of his scientific work. He spent an enormous amount of time dealing with the government, which wanted to tax church property, and he felt that the taxation was unconstitutional and unjust, and therefore refused to pay the tax at great jeopardy to the monastery and to himself. And to where this is one quote from the Lord Lieutenant of the municipality of Brno, who stated, physical measures would have been needed to put the warrant in execution, the prelate having declared that the municipal officers would have themselves to take the keys out of his pocket if they wanted to open the cash box, and would have to use force to remove any property for sale. That's how obstinate he was about this taxation of church property. He died on January 6, 1884, highly respected as an abbot and prelate. And also, interestingly, as a scientist. He is mentioned in his obituaries for his work with plants, but not as the major discoverer of the principles of inheritance. One of the young friars gave a very nice tribute to him, writing in Latin, but translated here in English. Gracious, generous, and affable to everyone, brother and father to us brethren he was. Flowers he loved, and defender of the law, he fought injustice. Exhausted in the end, he died wounded in the heart. And so with that, I'd like to turn my attention now to the other part of my presentation, which is the influence of Gärtner's book on Mendel, which is current research that I'm doing now with students and with colleagues. That this is the book that Mendel referred to more than any other in his classic paper, which is written by Carl von Gärtner and is a vast compilation of plant hybridization experiments and their results. And Mendel heavily annotated this book. He also referred to Gärtner 18 times in his classic paper, but interestingly, all but 4 of those references come in the concluding remarks section, which Dr. Van Dijk and Dr. Ellis have determined was probably written after his presentations, and oftentimes referring to species other than the pea plant. And in fact, when we look at some of Mendel's annotations, this is on the back cover of the book, you'll notice that everything there is referring to other species of plants. And yet there is this passage right here that refers to peas. And Robert Olby, looking at this, I'll just paraphrase what he said here, which is that he presumed that Mendel was summarizing passages in Gärtner and therefore exploring different types of things types of characters that he could study in pea. But what Dr. Van Dijk and Dr. Ellis have discovered is that most of the pea species mentioned in this passage do not appear at all in Gärtner, and that they only appear, in fact, in a flora that was written in 1863, which was after Mendel completed his experiments. So these notes probably do not apply to Mendel looking for characteristics to study in pea. He had already done his experiments by the time these came about. And Dr. Van Dijk's given me permission to let you know there is a new article on this that he and Dr. Ellis has been accepted in the journal Genetics and will soon be published. And in fact, here's that information. these are the references to Pisum in Gärtner's book on these pages. You'll notice this is Mendel's annotation right at the beginning of the book, Pisum on page 499. And yet these are pages in red are the ones that Mendel annotated. In other words, the vast majority of the more than 300 annotations that he made in this book are not in reference to the pea plant. Now, Darwin also had a copy of this same book, and he annotated it very heavily. In fact, even more heavily than Mendel did. And what's interesting is to compare Darwin's annotations with Mendel's in this book. Some historians have claimed that Darwin's ideas were so different than Mendel's that he would have had little interest in Mendel's experiments and theory. And yet when we look at the annotations of these two scientists in the same book, it's very clear they annotated many of the same passages, had very similar interests in them. This to me indicates that Darwin certainly would have had a great interest in Mendel's experiments had he known about them. Let me just show a few examples of this. This is one right here where if you read the translation of Gärtner's passage here, but up to now this is still to be considered as a special exception in Pisum sativum, so that we believed it to be justified in adhering to the law stated earlier, namely that the influence of foreign pollen has nothing to do with immediately changing the forms and external attributes of the fruit and seeds peculiar to the maternal plant, but that only in the embryo is the capability of producing through the germination and further development of the new plant a modification of the species concerned and the form of its parts. Now Mendel, that part in yellow is you can see Mendel's mark up here on the right. Darwin made a marginal note there stating it seems he does fully admit that artificial fertilization in Pisum affects seeds. The idea is that the artificial fertilization is not affecting the maternal plant, but only in the embryo, but that the seeds then appear in the pods. And this second passage is very similar. It reads in English, similar observations of different colors of the seeds in the same pod as above in Pisum sativum macrospermum and viridae were also observed by Professor Wiegmann in the second generation, with second generation in italics. Darwin's note, the peas in second or higher generations varied in color. Very Mendelian sort of approach. And then this is, I think, the most fascinating of the passages that we came across. Should this tendency of the hybrids to segregate their factors back and forth in subsequent generations not be taken as direct proof of inner necessity, independence, and peculiarity for the stability of the plant species, which refutes the stability of hybrids and their elevation of species? Here is Gärtner talking about the segregation of characters. He's referring to the color of pea seeds, and Darwin then annotated this passage stating, cause of variability lies in all generations. And so very clear, to me, this is very clear that Darwin had the mindset that would have set him, made him very interested in Mendel's work had he known of it. Now, also, it's interesting that there are passages in Gärtner regarding species evolution, which is a topic that Mendel addressed in the concluding remarks section of his paper. This one reads in a translation of Gärtner's German, of greater importance for our investigation of the nature of plant species is the a priori view of some natural philosophical botanists who accept the constant development of animal and plant forms as an axiom and infer from the perfectibility of man the changes in animals and plants. Friar Wimmer opposed this assertion from a philosophical point of view. Now, if we read from Mendel's paper, probably a reference to this passage, by the way, through the success of transformation experiments, Gärtner was persuaded to oppose the opinion of those naturalists who dispute the stability of plant species and assume continuous evolution of plant species. He sees in the completed transformation of one species into the other the unambiguous evidence that a species has fixed limits beyond which it cannot change. Although this view cannot be afforded unconditional validity, nonetheless, a confirmation deserving notice regarding the supposition made earlier about the variability of cultivated plants is found in the experiments performed by Gärtner. Now, some people cherry-pick this passage and claim that Mendel was stating that each species has fixed limits beyond which it cannot change. But when we look at it in the context, Mendel is taking much more of an evolutionary Darwinian approach here. And then here is a passage from Gärtner that in English translation reads, G.R. Trevihan states that the organism in living nature, like everything else that exists in space and time, is subject to incessant changes, that therefore the organization of animal and plant bodies also change, and whole species perish, and new ones arise in their place. You know, very Darwinian evolutionary point of view from Gärtner. And here is a passage from Mendel from one of his letters, in fact, his last letter to Carl von Nageli, where Mendel wrote, Gärtner was also convinced by his experiments that the male principle, as he put it, is always affected first. If this were actually the case, the naturally occurring hybridizations in Hieracium should be ascribed to temporary disturbances, which, if they were repeated or often or became permanent, would finally result in the disappearances of the species involved, while one or the other of the more happily organized progeny, better adapted to the prevailing telluric and cosmic conditions, might take up the struggle for existence successfully and continue it for a long stretch of time until finally the same fate overtook it. Interestingly, notice the passage, struggle for existence. Mendel's German words were Kampfdasein, which is the very words that Darwin used in the German translation of Darwin's struggle for existence. And Hugo Iltis, in his first biography of Mendel, wrote, the clericalists who stigmatized Mendel the liberal as a Darwinist were not so far wrong. So as a conclusion, Mendel's and Darwin's annotations in Gärtner's book offer an immense treasure trove of information revealing the contemporary thinking of two of the most influential founders of modern biology. And this is still very much a project in progress, and much still left for us to research on this. But I'm happy to present a little bit of that to you today. Many people to acknowledge on this, several of whom are present here in the room. But I'm very grateful to all of our collaborators in the work that we have done on this. And with that, I'd be happy to answer any questions.
So who can we give the floor? There are questions both from the audience and from the team's connections.
And by the way, I have a hearing disability. I have hearing aids, but I might need to have someone repeat a question for me if I have trouble hearing them.
Many geneticists who want to ask a question.
Here we go.
Yes.
Thanks very much. Perhaps the obvious question, what about genetic linkage? Was this never a problem for Mendel? Did he never see that?
I'm sorry, I didn't quite hear that.
Well, the independent assortment of traits doesn't happen necessarily if they're on the same chromosome, right? You have genetic linkage. Did he never run into this problem in his research?
Yeah, now Mendel, if I understand the question correctly, Mendel observed independent assortment for the 7 characters that he looked at. Of course, if genes are linked, then they do not assort independently. And it's some people have said, you know, is it that Mendel looked at 7 characteristics and there are 7 pairs of chromosomes in the pea plant? Was he lucky enough to just get 1 on each chromosome? No, he wasn't. Some of them are in fact located on the same chromosome, but are far enough apart that they do assort independently, because crossovers occur frequently enough between them. And but it's really not that surprising that the 7 characteristics he found did all assort independently, because for him to detect linkage in the type of experiments he was doing, the genes would have had to be fairly close to each other for him to detect a deviation from independent assortment. Is that the answer that you're? Yeah.
I have a question for you.
Sure.
So what is your hypothesis about why the discoveries of Mendel faded away in history for a while and then came back? Was that because of the success of Darwin, or was it the other explanation? Or?
Yeah, I think there's really quite a combination of factors that can explain it. It's not just a single thing. Part of it may have been that Mendel's publication came shortly after Darwin's Origin of Species, which by then, by the time Mendel published his paper, Darwin's Origin of Species had become very popular. And in fact, translations of it were being made in most European languages. And in fact, Mendel had a German translation of it, which was the second edition German. And so Darwin was really all the rage at the time, but also Mendel himself was reluctant to conclude that he had discovered something really outstanding until he could do additional experiments, which he did, but most of which were never published. And I think had more of those experiments been published, he might have been better known. Some have claimed that it was because he published in an obscure journal and was working in an obscure city. I don't accept that argument as valid, because the journal that he published in was routine for scientists at the time to present their results at the natural history museums or meetings of natural history societies. And in fact, Mendel's first two papers he presented at the Natural History Society in Vienna, and then later his famous paper was in Brno. But the journal was present in libraries throughout Europe and England and even in the United States, and so the paper was readily available. But not even Mendel, I think, recognized how important his discovery was. And it was only later, when others, in essence, repeated his experiments unaware of them, although there's even some question as to whether his rediscoverers truly were unaware of him. And in fact, Correns had even mentioned Mendel earlier. So but anyway, it was only in around 1900 when Mendel's results then became famous and Mendelian genetics was established. But in my opinion, I think it's a combination of factors rather than just one.
And how did he practically work? Because what can we learn from him nowadays? Because nowadays, if you do scientific work, also at KeyGene, at universities, you work in a team.
Mm-hmm.
And he apparently worked alone, or not?
No, he did not work alone.
Oh, okay.
These experiments truly were his, but he was very much collaborating with the other scientists in his area. And in fact,
and so on.
Several of the other scientists who were members of the Natural Science Society published hybridization experiments and even presented them. And in fact, interestingly, many of those were on Hieracium, the hawkweed, which Dr. van Dijk will be telling us about. But if you look at prior to Mendel's experiments, in the annals of the Natural History Society are reports of hybridization, both natural and artificial, in Hieracium. And Mendel contributed to Makowski's book that, we talked about Makowski presenting on Darwin. He was very much collaborative, and even the letters that he wrote to Carl von Nageli in Munich illustrate the collaboration that he was carrying on with a very famous botanist. One of the tragic things there is that the first letter that Mendel wrote to Nageli corresponds with the first letter that Darwin wrote to Nageli, and Nageli was corresponding with both Mendel and Darwin at the same time and never put the two of them together, which is unfortunate. But no, Mendel, this idea of an isolated monk who had plenty of time on his hands and was fiddling around with peas is very much a myth. He was a very collaborative scientist and worked closely with his colleagues, and even was elected as the vice president of the Natural Science Society later.
So you would encourage current scientists to do the same?
That's right. I would definitely encourage current scientists to work as a team.
Take one message, I think.
And
other questions, or check the Oh, I have
And I think my time is gone, but
I have a last question from
Oh,
the,
yes.
from the teams. Johan Volman, when Mendel was planning his PISM experiments, might he have already considered Gärtner's assumptions, or did he come across Gärtner's only later?
and best
By the way,
regards.
best regards from Vienna.
Yeah, and yeah, Dr. Fulman and I are very close friends. So yeah, might Mendel have it? That's really hard to tell because we don't know exactly when Mendel acquired the book. Now, he likely, I think it's more than likely that he acquired it when he was in Vienna. The book was published in 1849. Gärtner died in 1850. Mendel enrolled at the University of Vienna in 1851. Unger was a devotee of Gärtner's, so it's very likely that Mendel acquired the book while he was at the University of Vienna, and therefore that book may well have influenced his experiments. It's clear that he did mark passages on PISM in there. Whether he marked those before or after, we could never know, but very high likelihood, I think, that he annotated them before, even though many of the other annotations apply to other species that he worked with in subsequent years. Of course, he had the book throughout his life, and those annotations probably represent Mendel's lifelong reading of that book. And so it's my opinion that yes, Gärtner's work and some of the passages on P and Gärtner's book certainly may have influenced Mendel, and given their close association with some of his experiments, that may well be the case. But there's no way that we can actually know when Mendel read them and when he made the annotations.
Okay, thank you very much,
Thank
Professor Fairbanks. Another round of applause. So we make a big jump in time from the 1850, 1870 to 2023. And I would like to introduce Professor Dr. Manfred Keizer. He heads the Department of Genetic Identification at Erasmus University Medical Center in Rotterdam, the Netherlands. He is a professor of forensic molecular biology since 2004. He did his PhD in biology genetics at the Humboldt University in Berlin, Germany, on Y chromosome genetics and applications in anthropology and forensics. This has led to the introduction of forensic Y chromosome analysis, nowadays part of the standard toolbox of forensic DNA laboratories, crime scene investigators around the world. Professor Keizer's postdoctoral research focused on human evolutionary genetics, first at Pennsylvania State, USA, and later at the Max Planck Institute for Evolutionary Anthropology in Leipzig. And together with his colleagues, he discovered, for instance, the dual sex-biased genetic origin of Pacific Islanders in New Guinea and East Asia. In the Department of Genetic Identification in Rotterdam, Professor Keizer and his team perform fundamental and applied research utilizing the full range of molecular biology techniques. And over the last years, his team has made milestone discoveries in unveiling the genetic basis of human appearance and bringing this genetic knowledge and the suitable DNA technology they developed and validated into societal relevant applications, such as forensic casework. And today, he will start talking about his and his team forensic DNA legislations in the Netherlands and other countries. They were changed to legally allow the prediction of appearance traits in criminal casework, where it now routinely applied in many countries around the world. The title of his presentation is Human Appearance Genetics, Mendelian or not? The floor is yours.
Was embarrassing to hear about myself, but let me start with congratulating the ladies in the room. It's International Women's Day today, yes. And there should be a woman standing here. So when Peter asked me, I was first said, oh no, of course I can't come because what we do may not fit. So let me ask you here. So of all the appearance traits you see every morning when you look in the mirror, do you think any of those follows Mendelian inheritance? Yes, yes, yeah. Give me some. Eye color, you say? Okay. Hair color, you say? Okay. Nah, no, but that goes a little bit in a different direction indeed. Yeah, so that's of course one of the answers. But yeah, so is there more opinions about Mendelian traits when it comes to appearance? Body length, okay. Yeah, I think it may depend how you define Mendelian inheritance, but of course the typical idea, of course, is it's a single gene, single effect, Mendelian trait, familiar diseases, right? So is that applying to appearance? Well, in the past, people did think about eye color as Mendelian. And today, actually, it all boils down, it's not body height, to one, hair color, but only one, which is red hair. But only because it's one gene, but has lots of variants in that gene that all make hair color, but different red hair individuals have different of these mutations. Yeah, but red hair goes closest to Mendelian. Yeah, and so therefore I was a bit reluctant to come, but Peter convinced me, no, come and show, and here I show. And of course, there's lots of variation in appearance, and that makes me exciting working on it as a geneticist, but also as someone who wants to bring it to society as I come back at the end of the talk. It is almost individual specific. And of course, you know all of these guys here, the famous lookalikes, and the press likes to create the opinion that they are frequent, but in fact, they are very, very rare, and people have to look a lot. But of course, once you have DNA variants, this eye color, there is a bit of understanding, but much less. So the first part of the talk really is to give you an idea of how complex human appearance traits actually are, which simply is a number game based on genetic loci discovered. And when we first looked at this 15 years ago, a couple of years after I started in Rotterdam, on the simple example of eye color, indeed, it looked like Mendelian trait. And actually, this is a GWAS on very small number of SNPs, as you did at that time, on very small number of individuals as we had at that time, and you find a gene, yeah? Which means that this effect is very strong, that it pops up in only a couple of hundred individuals. Yeah? So you would think, well, that's the gene for eye color. That's the one gene for eye color. Right? Well, it's not, because you keep going and you say, well, hang on, it's not only 3 categories eye color. Eye color is a continuous trait. Let's do GWAS on continuous eye color. And you find more genes. And you say, oh, come on, 10,000. Ah, you have to do more because association testing. Well, let's do more. Let's do 200,000. And what do you find? Well, 60 genome-wide associated genetic loci for a trait like eye color on categorical eye color GWAS, where people in the past thought is not more than 1 or 2 genes. It's not. So why did people think it's only 1 or 2 genes? Why typically, if you have a brown eye and a blue eye parent, typically the kids are brown eye. If I have my family here, I'm blue-eyed, was blonde. The mother of my kids is brown-eyed, brown hair. Both of my daughters are blonde, blue eyes. So they would go against the norm, but typically it's brown being dominant. Why? Because there's one gene, and that's actually the gene we found first. That gene has a very strong effect. And because of that gene, you see this pseudodominance, but if you keep looking for genes, you find probably hundreds, all with very small effects. And that's a problem of complex traits mapping. And that's also the problem of appearance traits mapping. Lots of genes with lots of small effects. Okay, so hair color, very similar story. If you do large GWASs, as we now can do with 300,000, you find large number of genes. Obviously, they overlap. It's all pigmentation. It's all the same pathways, right? And there may be some non-overlap that may not be biological. So far, there's only one clear evidence of a pigmentation gene that contributes to 2 pigmentation traits, but not to another one. And that, again, is red hair. So the MC1R gene is causing red hair. It's causing pale skin, and it seems to have nothing to do with eye color. Whereas most pigmentation genes have effects on all 3 pigmentation traits, as you would expect. Skin color, similar. Obviously, skin color has a problem that you shouldn't do GWAS on skin color. Why? because skin color is a global trait, has largest differences between continental populations, which of course you cannot pool in GWAS because then you find all types of things that are between Africans and Europeans and Asians, right? So therefore people do GWAS within continents like Europe, where of course variation is small, so you only find a subset of genes. But still, it is dozens and will be hundreds if the numbers gets larger and larger. So this is a trait where people think, oh, what's this? Eyebrow color, right? Yeah. Sometimes you do what you can do, not necessarily what is most exciting. There is a large correlation, right? Most blondes have blonde eyebrows, but some blondes, as a kid, I always had non-blonde eyebrows. And I'm not blonde anymore. And I bet, although there's no scientific evidence yet, there's anecdotal knowledge, that this is actually the sign of you becoming brown in adulthood if your eyebrows were already dark. Yeah. So there is a strong correlation between head hair and eyebrow color, but it's not a complete correlation. Yeah. So you do find the pigmentation genes, as in this first GWAS ever. People are not so interested in eyebrow color, obviously. And there's one extra gene where we don't actually know yet if that's a real one. So if you move away from pigmentation, it becomes worse. Why? Because everything that is not pigmentation doesn't have these large effect genes. They seem to not exist. It seems to be all small effect genes. And then, of course, you need many more to explain the phenotypes. And that means you need larger and larger GWASs to find the genes. Here's an example of hair structures, whether the hair is straight or curly or wavy. Yeah. If you do 16,000, you find 12 genes. Eyebrow thickness, again, sometimes you do what you can do, not what you want to do. If the eyebrow is thick or thin, yeah, of course it's genetics, you find the genes. Face. If you map the face, you take distance between landmarks, you do GWAS, you find the genes. But the effects are tiny. And of course, with 13 landmarks as here, you're not characterizing the face in any detailed level. It's just very broad facial distances, right? So if you want to improve this, well, typically in the GWAS business, you do more samples. In the appearance genetics business, you can't do this because hardly people having those data, right? I mean, we are collecting images and DNA and do this, but not many people in the world do this. So if you can't enlarge GWAS sample size, the other way to do this is actually you have to work on your method. Yeah? So that's a new method just published a couple of months ago, where we tackled the problem of GWAS and complex phenotypes that consists of correlated subphenotypes in a better way of doing GWAS. That's a kind of combined GWAS approach. And if you do this and you apply this to the same data as the conventional GWAS on the lower side has just revealed 17 or so loci, you now find 50. If you have a phenotype that is composed by correlated subphenotypes, of course, the face is the classical example for that. So there are probably hundreds of genes out there that determine the face. And by far, we are not there yet. Okay, second part of the talk is, so GWAS is association. What I'm interested in is prediction. So I want to take a DNA variant. I want to tell you what color of the eyes there is for something I tell you in the last part of the talk. So here, the key thing, which you may not be too familiar, is a prediction accuracy value that takes specificity and sensitivity both into account, and therefore is more simple to explain. And that's AUC, right? That's what people use. So the thing to know is it doesn't run between 0 and 1. So therefore, you can't put it in percentage. It runs between 0.5 and 1, where 0.5 is basically random prediction. You can forget about your genetic data. You can toss a coin. And 1 is every time you predict, you're correct. So I show you a couple of traits and a couple of AUC values to get an idea how that works, right? So for eye color, blue and brown eye color only, it works very good, right? So you add the number of SNPs, you calculate AUC, and you're very close to 1 with blue and brown. And you're by far not close to 1 with intermediate. So what is intermediate? Well, everything that is not blue and not brown. Because in true reality, eye color is not a categorical trait. Eye color is a continuous trait, right? And actually, that's a problem because in practice, we cannot predict intermediate eye color by a high individual probability of a person having intermediate eye color. But what we can see is that people with intermediate eye color have similarly high blue and brown eye probability. And it's probably the reflection of what's going on. But at least for blue and brown, it works very good. So there's a test, you can apply it, NFE is applying more later. This is a little bit what these AUC values mean. So these are 40 people randomly picked from the department across the floor. And we run the test and we order the eye images according to the probabilities, with the highest probability image ordered left upper corner. And then you go down and ordering the images according to decreasing blue eye probability. And the highest probability for brown eye in the lower right corner. And you go up by decreasing probability. So what you should see is blue on the left, brown on the right, which basically you see. Right? And then you have these rare examples here, a person with 70% brown eye color, but obviously the eye color is not brown. So that's the part of the point something, 0.5 AUC, which is not correct. But for blue and brown, it often is correct. And then you have these guys here, which I mentioned, right? So this is an eye, which from the distance you will classify as this is not blue and not brown. Why? Actually, it has blue and brown components. And these are the eyes you typically classify as not blue, not brown. It's not the greens. The greens are so rare, even in Ireland they are rare, right? And here you see similarly high probabilities for blue and brown. Yeah, so there is a way to predict intermediate, but just not from highest probability point. So hair color works very good. It works best for red hair, but you have to have lots of SNPs of the MC1R gene in your test, because different red hair individuals have different MC1R mutations. So this test here has half of these SNPs only for red hair, which makes it difficult because red hair is rare, right? So it comes to the price of putting lots of stuff in there for something that is rare anyway. And why is this lowest? Because of people like me, right? I was white blonde as a kid. I'm brown now. Some people with a high blonde hair probability stay blonde and others get brown. And we don't know why that is. And we don't know why some people change from blonde to brown and others do not. So this, of course, is gene regulation, but we have no idea how that works. But that's the reason why, of course, the prediction of brown and blonde is least good, because you always have these people in your test set. So there's a test, and here are the examples, right? So you have these, but you have those, and if you look them up, they were all blonde as children. Skin color is interesting because most skin color genetic knowledge comes from Europeans, because that's where the money is. There's only little African GWAS on skin color, and still the variants we find predict most or predict best for dark. And this has to do with the evolutionary history, of course, of pigmentation. We were all dark skin in the past. So the ancestry variants are dark, right? So derived variants are light. And there's a test, and you can do this. And there's an example here, right? So you can guess who that is, a person with whatever, 55% brown hair, 40% blonde hair. That already tells you something maybe. 70% blue eyes, more. 66 pale skin, 25 or so intermediate skin. Goedemiddag. Okay, what else can you predict? Eyebrow color, well, not so good, right? I mean, AUC 0.75 is the middle between random and completely accurate, right? So this is where it starts to become interesting for practical application. Yes, you are there for blonde, you're not there for the others. Red eyebrows are not in there because they're so rare. Even in this sample of a couple of thousand, we didn't have them. Hair structure, this is only straight or not straight, you can predict. AUC of in the area of 0.7, yeah. So this is all examples where prediction starts, but it's not there yet. Male pattern baldness, largely genetics, hundreds of genes. If you predict, well, depending on what category you want to predict, there's a variety of AUC. You just want to predict any versus no, it is not so bad. It's in the area of 0.7, but you need lots of SNPs. Body height, you can forget about it if you want to predict normal range body height. The shortcut here is predicting tall stature. If you work in the Netherlands, you can do this. You can enrich your population sample by hundreds of extremely tall people because they are there. They're happy to volunteer, right? 0.8, but with 700 SNPs, right? Which is not so easy, as you all know, to put in a simple DNA test, right? So that's the problem of predicting traits that are caused by genes with small effects. You have lots of these DNA variants, and if you want to predict a practical application, you have to put them all in a test, and then running samples that are bad, as I come in a minute. Face? No way. Why? You see it here. So you do GWAS in 10,000, you find whatever, 20 significant loci, you take the SNPs and you quantify how much do they explain of the facial variants. And this is simple 13 landmark 78 distances. They all together explain less than 5%. So you can forget about prediction. I know there's one company that sells this. You can forget about prediction. So now the prediction. So I haven't published in Science, but Science published about me. 2011, they wrote a 2-pager on our work in Rotterdam in a series that was about the anniversary of the human genome, because they figured, or I would agree, maybe others not, that at least this application of the human genome to put this into crime scene solving tools obviously is a good one for society. So why do you want to do this? Well, the trouble with DNA identification is you can only identify someone you already know, because the trick you do is simply comparative. DNA profile from crime scene sample has to match the DNA profile of the reference sample of the person you want to identify. No match, no DNA use in forensics, right? So there is a database of criminals, and if that person gets out of jail, commits a crime again, the DNA profile stays in the database, the next case, boom, match, you have him, right? But the one murderer, like the Fadra case, who never murdered again for decades, no way that you will find him with a simple matching. So cases like Fadra, no match, no other information, that's it for DNA, right? But if you can predict how a person looks like, where the person comes from, how old the person is, in a field that people nowadays call forensic DNA phenotyping, you can tell the police, don't look for anybody, look for this group of people that match these characteristics. So does it work? Are cases solved? Well, the Melieke van Doorn is one of those cases. The Fadra case is not the very good one, because there indeed people didn't do appearance. At that time, nobody could do appearance prediction except red hair, but they did ancestry prediction. And of course, in Friesland, with a prediction of a European ancestry, which was done at that time, you don't come very far, right? And therefore they had to do 8,000 until they found the guy, but only because he participated, but they would have found him anyway. So this here then was the shortcut, right? Because there they did ancestry testing, also appearance, but that's not in the press. And they found Turkish ancestry in Zaandam. Obviously, this is more leading because it's not the majority population. If you read this all there, so if they combine forensic DNA phenotyping with familiar search, so you do DNA profiling not on the autosomes, but on the Y chromosome, with the advantage that if the perpetrator doesn't participate, which in this case he did not, you will find him as soon as a male relative participates, because the male relative has the very same Y chromosome. So here, they only focused on the 135 Turkish ancestry people who lived in this area of Zaandam where the murder happened at that time. And they only asked those for voluntary participation. And all but 2 agreed. And they found a Y match, but no autosomal match. So they knew it's not the guy. And then police investigation showed that one of the 2 non-participating was the brother. And then it was the court order. And then, of course, it was a DNA. So this is how things can go if you are lucky. Well, obviously, you have to be lucky, and you have to do more than appearance and ancestry prediction, where this familiar search on the Y chromosome is a good one. So you have to combine things to be effective. And then 2 last examples. So you can also apply this to anthropology or human history studies. This here is King Richard III. So Shakespeare was right. Huh? So he was a hunchback, right? So actually, it's a nice story. It's a Nature communication, but you should read that. So they digged him up in Leicester City underneath the parking lot. And actually the feet you don't see here, the feet are missing because there was a building there from the Victorian times. Yeah? And if they would have built the building 2 meters here, he would be gone. Yeah? So obviously they did all types of DNA, full mtDNA genome matching with the maternal relative. So we said, well, let's look at eye and hair color at that time, which we could do. So very high probability for blue eyes, fairly high probability for blonde. But of course, we know the problem with the blondes, right? So these are blondes that have, or browns, blonde kids that have these type of high probabilities, right? So obviously the conclusion then is, whoa, he most likely has blue eyes, and he either was blonde or was blonde as a kid and became brown later on. And then of course you say, why are you doing all this? Look at painting, right? So even for a king, there were no paintings done while he was alive. So all of these paintings were done post-mortem, and you see him with all types of eye colors and hair colors as the artists have remembered. Yeah. So that's the one that showed him most closest after his death. Yeah. And that's probably very close to reality. Yeah, so he probably was like me, blonde, but then getting brown and with blue eyes. And there's another example. This is Cheddar Man, who lived 11,000 years ago in Britain. Yeah, digged out of Cheddar Gorge, somewhere in England. Who became famous of one of the Mesolithic individuals in the UK. And we were involved to doing eye, hair, and skin color prediction. So these are the predictive values. And what is striking here, of course, is the discrepancy that you would not expect between light eyes and dark skin. And obviously this then hit the press and everything, right? And it's still puzzling if that's really true. I mean, we believe the prediction because it was good sequencing data, it was good sequence coverage, which always is a problem with ancient DNA studies because they never do targeted, they all do genome-wide, and then you don't have good coverage for the SNPs you're really interested in prediction. But we know at least 1 or 2 other individuals from the Neolithic and Mesolithic times that also had light eyes and dark skin. And why is it unexpected? You're looking at the watch, I'm done in a minute. Because the idea was that actually it was skin color that evolved first, because skin color has the environmental interaction, right? It's the UVB pathway that with the dark skin and African origin, you migrate north, much less UVB, you suffer from your dark skin because you get some problems, even birth problem with bones, because you basically overprotect, you don't have enough vitamin D, right? So therefore there was a trigger to lose the dark skin to become light. And that evolutionary adaptation thing does not exist for eye color and hair color. So why then light eyes and dark skin? Ooh, we don't know. So my gut feeling is this simply is a sampling artifact. So if I mix African and European, so this is about going back to Mendel and doing mixing experiments in humans, huh? Right. So if you mix Africans and Europeans, and of course these populations are there, African Americans, right? So in more admixed African Americans, you do see these phenotypes. You do see dark-skinned people with light eyes, right? But of course, you also see African Americans that have dark skin and dark eyes, and you may see more European admixed African Americans that have light skin and light eyes and light skin and dark eyes, right? So let me sample 1 from a pool of thousands African Americans. Well, there may be a good chance that you select a dark skin, dark eye, but there may be also a chance that you select a dark skin, light eye, right? And maybe the population at that time already, and we're talking 11, 12,000 years ago, was already more mixed in these phenotypes. And by chance, so far, we run into these guys and we haven't run into the other guys. Okay, and that's it. These are the people that did the work with me. Here's where we got the money from, not enough, but that's what it is.
Well, thank you very much for a fascinating presentation. Questions? Yeah.
I'd like to challenge you on your sampling artifact for the skin color, because there was a recent paper showing more Mesolithic genomes which had a dark skin prediction. You don't agree with those results? 1. But they're sort of So that's 2 out of 3. Ain't bad, right? But I understood that they were thinking more in terms of diet being the driving factor, because these people had a more vitamin D rich diet. And when farming came, that's when the selective pressure really kicked off.
Yes, you can compensate, of course, if you eat fish. That's basically it, right?
And then the final, do you think we should use these predictive phenomics for criminal investigations?
Oh, we do,
But do you think we should?
of course.
And what if you make the wrong prediction? I mean, I wasn't so
So what if you
impressed
make
by
the wrong
some of those.
prediction? So here's the example, right? So what people do nowadays, they use phantom drawings, right? So there's quite some scientific evidence how wrong human memory is and how much that reflects in phantom drawing, right? The trouble is, if you do a phantom drawing in one case, you have no idea what the error is. So if I predict eye color with 95%, obviously you can argue about how good my model is and samples and everything, right? But I know the error is 5%. If I predict eye color with 80%, I know the error is 20%, right? So the idea actually is to educate police to use this in a more intelligent way, actually using these numbers, using these errors, and basically saying, well, if you have 95% eye color probability, you better look for a blue-eyed person first. If you have 80%, well, you do look for a blue-eyed person first, but you don't look forever because you have a 20% chance that it's wrong, right? So if you do it right, I think, yes, you should do it, but you should do it after science and technology allows this and not before. And the face example of this company in the US is very much before, and you shouldn't do that.
So we have a question from the Teams connections. Mohammed Farouk, how does the evolutionary distances could affect the predictions?
I don't know what evolutionary distances means.
Farouk, can you clarify a bit?
So let me answer it differently. So if I take the Neanderthal genome and I look at our sites, I predict dark. So does it mean they were dark? No. Because I base my knowledge on modern populations, right? So if by chance these Neanderthals were light, but involving other genes than are in modern populations, you don't know. How likely is this? So there's one example of not the same genetic base. Blonde hair, you would say, is what? A European phenomenon. But indeed, so there are blonde hair people on Solomon Islands and Papua New Guinea who are as blonde as Europeans, but have pitch black skin and pitch black eyes. That's a nice one on the correlation of the genetic correlation. So it is the same gene, but it's a different mutation, right? So in our prediction, of course, we use mutations, we use SNPs. And if in this case, we would predict wrong. Right? So is this evolutionary distance? Well, so if you go back in time, right, and you do prediction on ancient modern humans, well, you may think, well, you're safe on modern humans. But if you go to the Denisovans or Neanderthals, maybe not. Right? So they are the kind of ancestral prediction, meaning ancestral phenotypes, may be true or may not be. I would still say it is true because the chance that this evolved so different in the human lineage after they're separated from Neanderthals is pretty small for these traits. But yeah, with the blonde hair, it happened.
So Mohammed reacted that was the question.
Right.
So, Jeroen.
I was wondering about predicting complex traits. So it doesn't seem to work very well. What do you think you need to,
For others not?
what do you think you need to do to prove that? Or do we have to learn to live with it?
Well, I mean, it's genomic risk score discussion, right? I mean, that's what people do in medicine, a genomic risk score, right? So how do you get a better genomic risk score? You do larger GWASs because you need more loci, right? So depending on how complex your trait is, you first have to understand your trait. If you know all the genes and you put them all in your genomic risk score, you are done for the genetic and inheritable part, which of course for appearance is very large and maybe much larger for many of these common disease traits.
So you think it's just a matter of scale?
Yes, because monozygotic twins look the same for a large part of their life, right? And when they start to look different, it's age, it's environment, it's those type of things, right? So it is in the genes, and it's just our limitations to find it because it's so complex.
We have two more questions. One here from Peter.
Thank you for a very nice talk. I have a question about in plant breeding. We look a lot at quantitative trait loci. So how's that in human? I got a feeling that there are core genes, and then probably these extra ones are more or less regulators, the small effect. For instance, also you mentioned earlobe. I wondered if there's a QTL for that or something like that. Is there a major gene affecting it?
No, so the only appearance genes where you have major effects is pigmentation. Actually, you're saying regulatory. So the one gene you saw first, yeah, in the GWAS of 300-400 people, yeah. So that hits the OCA2 gene, which is a gene, if it's mutated, gives you no color. It's albinism. But this is not where the association signal. Association signal comes from it next to it. Why? Because there's a regulator. Very strong regulator that is behind that largest pigmentation effect that exists. It's not a very strong regulator. So there are lots of these regulators, of course, with beauty. So it's very hard to know what really comes from what part. But we don't see these large effects.
So last question before the pause.
Yes. My question is, does the Dalton phenomenon exist?
I'm sorry, I didn't.
The Dalton phenomenon, does
yeah.
it exist? Because I used to have a family in our hometown where they entered into church, and there were 5 children, and they were exactly the same. It's almost like twins.
Or the Dalton? No, what do you mean?
No, they were different in age, but they looked really alike. So like the Daltons in Lucky Luke.
Oh, yeah. Well, I mean, obviously there are some siblings that look more alike than others, right? And so why is that? Well, I would say because the parents must be more looking like another, right? Because if you have that and you do breeding experiments, obviously you don't end up very different, right? But of course, if you do different, then you do end up in F1, F2, whatever F. Some may be more different, right? Maybe there's more than that, right? I mean, we have no idea how this is all controlled, right? But yeah, it's obvious you have some siblings that are more similar than others. Yeah. I have no idea why. I would bet because the parents may be more similar.
So thank you very much, Professor Kaiser. And I would like to give him a big hand of applause. And I would like to thank both speakers for introducing us in this wonderful world of genetics. And I'll see you back after the pause at 15:40. 10 minutes past half. We're all back. So thank you for coming back, and we'll continue with the program. It's an honor for me to introduce to you Dr. Jiří Mitáček. He is the head of the Mendelianum and of the Department of History of Biological Sciences of the Moravian Museum in Brno, Czech Republic. Dr. Mitáček's team engages in interdisciplinary research in the field of history, philosophy, and methodology of life sciences and genetics, with an emphasis on the study of the life, work, personality, and nature of the discovery of Johann Gregor Mendel and other historically related developments in knowledge in this area. The Mendelianum has been systematically researching Mendel's legacy for 60 years. In addition to Mendel's work and personality, the team studies interpretations of Mendel's discovery in relation to evolutionary theory. The team also studies the history of biology and genetics in Czechoslovakia, especially on the direct and indirect influence of politics on biology and genetics in the 50s and 60s. And of course, also the latest developments in the field of molecular genetics, genomics, genetic engineering, medical genetics. They are also within the scope of the work of Dr. Ekara. So the floor is yours, and the title of the presentation is Look into the History.
Thank you very much for introducing me and work of our team. And it's a, I would say first that it's a great, really great pleasure to be here. And we are very pleased that Peter accepted our award. And because perhaps you didn't heard anything about Mendel Medal until today, so I will try. Let me introduce some. Let me get some small or briefly excursion to the history of Gregor Mendel Memorial Medal. So we are from Brno, you know. This is Mendel's city. This picture is very old. It's from half of 17th century, probably during the years of 30 Years' War, and the inscription is, and after 200 years after Second World War was established the Moravian Museum. This is the second largest museum in our country, and it was founded in July 1817 by the decree of Emperor Francis I as a part of the Moravian and Silesian Agricultural Society in Brno. Agricultural Society was very important for the founding of modern sciences in our country, especially in Moravia and Silesia. So this is the bishop's courtyard, the seat of the Agricultural Society. And Mendel was a regular member of this natural science section from January 12, 1855 until his death. This is the place where Mendel was very inspired by his colleagues and get some inspiration, and it was a place of exchange of scientific knowledge in those times. So today there is the Mendelianum Center, our center of the Moravian Museum. So this is the also greeting to Wageningen from Brno. In 1965, in occasion of 100th anniversary of publication of Mendel's discovery, a discovery paper, a large international symposium was held in Brno. And in this occasion was Mendelianum as a Gregor Mendel Memorial of the Moravian Museum opened. The symposium was held in August under the patronage of the genetics section of International Union of Biological Sciences, UNESCO, and the International Atomic Energy Agency, and others, and went down in history as a rehabilitation of Mendel's work after the post-war suppression or repudiation of genetics in the communist Czechoslovakia. So this is the very important time for the history of science in our country. This is the opening ceremony of Mendelianum in July 31. And this is the Gregor Mendel Memorial Medal, which was especially created for the Moravian Museum on this occasion in the year 1965. Memorial Medal is annual award given for development of Mendel's scientific legacy and for the support of the Mendelianum of the Moravian Museum, and has been awarded occasionally since 1965 and annually since 1992. So after the so-called Velvet Revolution, it's another great change in our country. This is the obverse of Mendel Memorial Medal. This is Mendel from the profile. It's interesting because this is a little bit fiction for the artist who created this, because we have no photography or documentation. Mendel is always from Anfas. So this is a little bit fiction, but I think that it's very, very, very great, good. This inscription, Gregor Mendel, of course, and reverse. Around this inscription, Amicis in Latin, in Amicis Autoribus Museum Moraviae, Brunense, 1865 to 1965, which means friends and supporters. And you can see the bucket of the stems and leaves of pea flowers, Mendel's experimental flowers. And on the belt, maybe paper, is Mendel's signs of dominating and combination of hybrid and homozygotes, and signature, of course, and dates. The creator was Václav Adolf Kovanič. This is a great Czech sculptor and artistic creator of medals, born in Polish Silesia in 1911 and died 1999 in Moravia. He studied Academy of Arts and Architecture and Design in Prague and Academy of Fine Arts. And he's author of 100 medals, and his medal work is debated to the important Czech and foreign artists and scientists as Mendel, Evangelista Purkyně, Comenius, or Leoš Janáček, and others. And largest part of his work of art is stored in our museum. And this is the list of holders or winners on Meridian from the bottom to the up to and to the top. And the first who was awarded, it's a lucky coincidence because it was the Hans Galliard from Erasmus University from Rotterdam, great human geneticist and very known communicator in science, especially here in this country. And you can see also, for example, important names like Franz Weiling from University of Agriculture in Bonn, Francisco Ayala from California, Irvine, Valery Soifer, Jan Klein from Max Planck Institute. And on the top in 2006, holder of Nobel Prize, Marshall Nirenberg from Bečeda. He was awarded this year. And in 2008, James D. Watson also visited us and was awarded of this medal. Also, 2017, Professor Fairbanks is also holder of Mendel Memorial Medal, and today Peter van Dijk from KeyGene. Wageningen, sorry. The taking over of this medal is traditionally associated with the Mendel, so-called Mendel-expert since 1992. This is an opportunity to present research rates of this medal. So this is some pictures from this Mendel's lecture in Brno. Professor Soifer, Professor Klein, Marshall Nirenberg, David Lane, James D. Watson, his partner, Professor Fairbanks. This is the last one from last year, Professor Uwe Oswald. And maybe you know this person, Peter van Dijk. So that's all what I very briefly informate about history and importance of this award.
Thank you very much.
It's a pleasure.
Any quick questions about the medal or not? Probably it was enough as an introduction. Thank you very much. Then I would like to introduce Dr. Jiří Mitáček, Director General of the Moravian Museum in Brno, Czech Republic. Already started to be involved in the Moravian Museum during his postgraduate studies in historical sciences in 2005. So that's quite some time ago. Moravian Museum is a regional museum. It offers school programs and surprising and attractive exhibitions, such as the World of Czech Omics. Czech Comics. Hey, is that correct? Is that not a typo? And about ancient jewelry and body ornaments. And of course, the museum harbors the Mendelian Visitor Center. It offers an attractive insight into the field of genetics and molecular biology to students and to the general public. Thanks to the combination of historic premises where the founder of genetics, Johann Gregor Mendel, worked and the state-of-art laboratory, visitors can actively try out a number of methods and laboratory procedures, from pollinating pea flowers to studying gene expression. And I would like to ask you now to come forward and to start the ceremony, probably.
So, dear colleagues, honored guests, Peter van Dijk is a plant geneticist who studies the genetics of clonal seed reproduction in plants, apomixis. He uses the common dandelion as a model organism. He started this research at the Netherlands Institute of Ecology and continued it in KeyGene. After his pisum experiments, Mendel worked on hawkweeds, which were later found to be apomictic. Van Dijk's dandelion and Mendel's hawkweed are both apomictic and closely related. In 2007, Van Dijk co-authored a review of apomixis. The title Mendelian Genetics of Apomixis in Plants indicated that apomixis itself was inherited in a Mendelian way. Thus, there was no fundamental difference between the genetics of Mendel's peas and hawkweed. Ultimately, Van Dijk's work at KeyGene contributed to the identification of the parthenogenesis gene in apomictic dandelions and hawkweeds. This is an important step in utilizing apomixis in future agriculture. In 2016, a conference was held in Brno to celebrate 150 years of Mendel's pea publication. On the recommendation of the pea geneticist Noel Ellis, Van Dijk gave a presentation about Mendel's hawkweed experiments. For that presentation, Van Dijk studied Mendel's letters to Nageli in detail. This revealed that much of the existing literature on Mendel's hawkweeds was incorrect. The result was a joint publication in the journal Genetics entitled The Full Breed of Mendel Genetics. The collaboration between Van Dijk and Ellis has been very fruitful. It has led to 7 other publications, ranging from Mendel's scientific work to his extensive international travels. In it, they make extensive use of newly available digitized sources and reinterpretation, biological knowledge, and digital archive work have contributed to a clearer image of Mendel. So for these reasons, the Mendel Memorial Medal Committee, which is a part of the Mendelianum of the Moravian Museum, decided to award this year's prize to Peter van Dijk for his research on the life and work of Johann Gregor Mendel. So
I have to work for.
Okay, I really feel very honored to give this Mendel lecture today. It's really something that has become out of control hobby. So I really feel very much really attached to Mendel. So today I'm going to talk about the shifting appreciation of Mendel's constant hybrids. And we'll give a mix of historical misunderstanding, the molecular characterization, and finally to the future agricultural application. Okay, like most of you, I encountered Mendel first at school, and it made quite an impression on me. Also, I kept my school book, so after 40 years when I looked at them, I still remember there was this woodcut of Mendel, and that actually it was the only scientist that had an illustration, although it was a woodcut and it was a little bit mysterious. But what it says that Father Gregor Mendel had a brilliant idea to study how traits are passed on by experimenting with plants that only differed in a single trait. And he published his experiments in a local scientific journal in 1865, but they were not giving much attention. However, in 1900, 3 biologists, de Vries, Sermark, Correns, arrived at the same conclusion as Mendel. And that really fascinated me. I remember that when I was at secondary school, and when I opened these books after 40 years, I found a scratch note. It's not very, well, it's not very elegant, but basically this is Mendel's laws, and it's a trifactorial analysis. There's you can see segregation, recombination, you can see dominance. So I was really impressed by that story of Mendel, and it's also characterized by the historian of science, Jan Sapp, and he said there's no greater legend in the history of science than that of the experiments of Gregor Mendel. That's quite a big thing. So what is a legend? You can think of a legend like the legend of King Arthur. So these are really catching or nice stories that we all like to hear. But the question is, is it true actually? What is true? What is really the truth in the story? And I got interested in that. So just to recap a little bit about Gregor Johann Mendel, you heard that before today. Here we see Mendel, there we see the monastery. So he was an Augustinian, he lived in an Augustinian monastery in Brno. Now it is called, so in those days Brünn, and nowadays called Brno. He was a friar. So a friar is not a monk, but the friar is a monk that is also having societal duties. So he was a teacher at secondary school in biology and physics. And he made his B crosses from 1856 to 1863. And he also, it's quite typical that he had conflicts with the authorities. So it's not only about the tax with the state, but he also had conflicts with the church. So you could say that he's a kind of a rebel, which makes him quite interesting. And he made these large travels to France, England, Italy, and Germany, which we only discovered exactly how they went just a few years ago. And then what is very important, after his death, so he died in 1884, then his notebooks were burned. So that is a big problem because we don't have a good historical sources to go after. So he only wrote 2 big major publications, also 2 tiny, but the important ones are first a large paper on peas in 1866. It's called Experiments in Plant Hybrids. So here we have the peas. And then there was a preliminary report on hawkweeds. And you heard that before, that hawkweeds was another species that Mendel worked on. I'm going to tell more about it in this presentation. So the English translation is on Horatio hybrids obtained by artificial hybridization. So only 2 publications. And so that's a picture of the hawkweed. So you can see that they have completely different flowers. And that's also an important aspect for the rest of the presentation. So when you compare that with Darwin, for instance, so here are a couple of my books that I have, my Darwin books that I have. And also, so it's probably half of what Darwin published, probably even a third or something like that. And also includes Mendel's publication. So in case you hadn't noticed it, so Mendel's publications are on top. So actually, it's a very thin top section. So that's what Mendel published. Well, in the history, we have more of these sudden geniuses. For instance, when you go to art, you can think of Johannes Vermeer. There's now a big exhibition in Amsterdam, which unfortunately is sold out. So he's very popular, he's very special. But we know so little about him that he's being called the Sphinx of Delft. So, and we also have, for instance, William Shakespeare, who is the greatest playwright in history. But actually, we know very little about this man, and even some people say that it was a pseudonym, and actually he was the Earl of Oxford, Edward de Vere. So we know little about these people. So when we go back to Mendel, so what made Mendel unique in the understanding of inheritance of the 19th century? So why was he the only person that understood genetics or understood inheritance? And how did he make his discoveries? And what did he think he had discovered? Did he really realize what he had discovered, or was it something different? And also the question that has been asked here before, why was he misunderstood? There are many questions there. So we see it in the school, probably with Darwin. He's the most prominent biologist from 19th century, but we know there are many uncertainties actually. So it has been mentioned before that I work on dandelions. I do similar work as Mendel did with hawkweeds. So in 2016, there was a conference that was celebrating the 150 years of Mendel's pea publication. That was held in Brno. So you can see a picture of that. This is the monastery with Mendel. And I was invited to give a presentation over there. So I gave a presentation. And when I had to prepare that presentation, I thought it would be good to go to the primary literature and to have really a close look and to read the primary literature, because often what we do is that we just in beans. So it suggests that Mendel was basically a plant breeder, and the impression that we got is that he started as a plant breeder, then he observed certain patterns in the inheritance, and then he really became curious and he wanted to find that out. So the idea is that the purely scientific research emerged from a plant breeding applied research. And we wrote a paper about that in genetics. That was called How Mendel's interest in inheritance grew out of plant improvement. And yeah. Okay, how about the peas? Peas you could call a variable hybrid. So when you think back at school, then you got this cross between two different pea colors, two different flower colors. And then you get, so the homozygous, the pure breeding, and then you get F1 hybrid, which is homozygous. And then when you do self-fertilization in the F1 hybrid, you get a F2 hybrid. What you see is that you get the white color back again. So it's in 1/4 of the plants is white again. So you could call these variable hybrids, because the hybrid, when you self it, you get variation in the offspring. So this is just to summarize that. The F1 hybrid is uniform, the F2 segregates, in this case in a ratio of 3 purple to 1 white. Now we go to the constant hybrid. So we go to the hawkweeds. So the hawkweeds actually were carried out over quite a long period of time. So when Mendel ended in 1863 his pea experiment, then 3 years later he started with his hawkweed experiments, and they lasted until 1873. Now, what is very important is that some so-called the Mendel-Nageli letters. And as I mentioned, we know very little about Mendel because all his notebooks were destroyed when he died. But in 1905, so many years after Mendel had died, 10 letters were discovered by accident, as it was described by Karl Correns, who was one of the rediscoverers of Mendel's work. And these letters were addressed to Carl Nageli, who was a botany professor in Munich. And they were written between 1866 and 1873. So indeed, many years after they were written, they were found back. And also fragments of Nageli's letters in reply to Mendel were found back in Brno. And what is very important is that these letters that they gave an insight in the way how Mendel was thinking. And they were mainly about the hawkweeds. Now, when Mendel published his P paper in 1866, he got reprints, and then New Year's Eve of 1866, he sent his first letter to Nageli. He had got these 40 reprints. One of them went to Carl Nageli in München, and he wrote a long covering letter. And just a little bit about Nageli. Nageli lived from 1819 to 1891. And he was considered to be the greatest, by many at least, the greatest botanist in Germany. So he was an important person. And this is a quote from Darwin. So Darwin considered him, or at least many considered him the greatest botanist in Germany. And he was a hawkweed specialist, although he did many other things. And what's important is that Mendel, in his first letter, he described that he already had crossed the hawkweeds. And he asked Nageli for help with determinations of these cross products. Now, in the past, it has been thought that these hawkweed experiments were very frustrating to Mendel. And the reason for that was that there's this idea that the hawkweeds were carried out to confirm the P results. So these are a number of quotations from literature. So one of them is Mendel used hawkweeds to verify the laws of inheritance he discovered in Pisum. And you can find many more of these statements. And then, of course, that was very frustrating. And people think that repeat his Pisum experiments failed, and it led to utter frustration, great disappointment, frustration, life's great accidents, frustrating attempts, frustratingly. And the idea is that Nageli actually suggested to Mendel to work on that. So we have a legend, which is nice. And we also have a bad guy in the legend, you can see here. But actually, Mendel himself had already started with making these crosses before he sent the first letter to Nageli. So what is important, and which is often not printed, so when you have the P paper, there's a long section which are the concluding remarks, and that section is often not printed because some people say that it might confuse students. So it's a little bit, yeah. But when you read that section, which often so is not there, then you can see what Mendel says is that he says that we encounter an essential difference in those hybrids that remain constant in their progeny and propagate like species. And he also says that this is of particular importance to the evolutionary history of plants because constant hybrids attain the status of new species. Well, this is exactly what he was looking at in Hieracium. However, when we go to the first letter of Mendel to Nageli, he describes his results in Pisum, and then he also describes how he could test his results in Pisum in other species. So he describes, he says, writes, in order to determine the agreement, if any, with Pisum, a study of those forms which occur in the first generation would be sufficient. So what he's saying is it would be very easy to test the Pisum experiments. And then the next sentence is It's the start of a new paragraph, actually. Hericium, so that's hawkweed, and Cercium and Geum I've selected for further experiments. And he also says last summer I tried to combine several Hericium species. So this proves that he already had done the crosses in Hericium before he sent the first letter to Negley. But if you read this, it looks like he used the hawkweeds to indeed to verify the results he found in Pisa. And this is an example of Geum, one of the species that the constant hybrids that he mentions here. However, when we go back to the original handwriting, what we see between pages 3 and 4, actually that these two, so where he describes the results in peas and how they can be tested in different species, other species, we see the hawkweeds, the thistles, Cercium, and the Geum. That is actually on a different page, and it's a different paragraph. So what could be the case is that there's a missing page here because these letters were discovered so many years after they had been written, and they were discovered by chance, which is also not very strong. So the idea is that there could be a missing page, and then the question is what could be on the missing page. Well, actually, in the next letter, Mendel describes his crosses with many other species, which actually are variable hybrids. So this would be very logical if the piece of the variable hybrids would succeed the part of the variable hybrids in Pisa. And then maybe at the end of that missing page, there could be something of the constant hybrids that has been described in by others in other species. So we think that can explain it. And then Mendel found his first constant hybrids in hawkweed. So in November 1867, he writes in one of his letters, whether the seedlings will retain the characteristics of the hybrids or whether they will show variation will be determined next year. I'm impatiently awaiting for the coming summer. So he's really curious. And then next year, April 1868, he writes, about 100 have overwintered. This far, these plants are indistinguishable from each other and agree with the hybrid mother plant. So this looks like a constant hybrid. And then after they have been flowering, or when they flowered, the first generation of last year's hybrids, consisting of 112 plants, is flowering. As far as I'm able to judge, all plants are alike in the essential characteristics. So what did he see? He made a cross between two different hawkweeds. He got a F1 hybrid. And then he got seeds from that F1 hybrid. He got F2 seeds, and they were all identical to the F1 hybrid. So you can compare this with the variable hybrids in Pisum, where we had 1/4 of the white parent coming back. But here you see only in the progeny of the hybrid, here we see constant hybrids. So they are identical, they don't segregate. So F1 is uniform, the F2 is identical to the F1, and there's non-variable. Now, Mendel had to make a lot of emasculations for this because he thought that he got a lot of self-fertilization. Later on, that turned out to be wrong, but he thought that he had to emasculate, so to remove the pollen of all these flowers to prevent them from self-fertilization. And this is a flower, actually it's an inflorescence of this is a dandelion, but the idea is the same as a composite species, as a hawkweed. And you can see that there are many of these small florets on receptacle. So this is a picture of a receptacle of a floret, and one single floret, and you can see the anthers. For the same size, there's a head of a matchstick. So this is really small, and he used a needle to get rid of the anthers and to get rid of the pollen, in order to to prevent them from self-fertilization. And of course, you need a magnifying glass or a microscope for that. So this is the microscope Mendel had. And he used a lens to concentrate the daylight, and he also used a mirror. So maybe you can predict what happened after maybe a month. He got a fatigue and exhaustion of the eyes appeared and reached a serious degree in spite of my immediately sparing my eyes as much as possible. And it made me incapable of any activities well into the winter. So this started in summer 1869 and went on well into the winter. So it was an incredible amount of work that Mendel did. And then to his surprise, in the beginning, he had only found one constant hybrid, so one hybrid type, but then he found several ones. So as a matter of fact, variants appeared in all those cases in which several hybrid specimens were obtained. And I must admit to having been greatly surprised to observe that the results diverge even greatly different forms. And I admit to you, honored friend, so he says to Nageli, which is one of the most important professors in Germany, honored friend, quite a friend, how greatly I was deceived in this respect. So what did Mendel see? So this is just of course cartoon. He did not get one constant hybrid or one hybrid, he got several of them. And these also turned out to be true breeding. And that was really puzzling to Mendel. This was what he saw before, only one F1 hybrid. And he sees a constant hybrid. So when you look at this, you see the F1 hybrid should be variable, and it segregates in different constant hybrid. So then we go to the end of the hawkweeds experiment, which is in November 1873. And then Mendel writes the hawkweeds have withered again without me having been able to give them more than a few hurried visits. And I'm really unhappy about having to neglect my plants so completely. So this does not fit very well with the frustrating experience. He's really unhappy. So if we summarize the results of Mendel's peas and hawkweeds. So the peas are variable. So in the peas, we see that the F1 is uniform, but the F2 is variable, the next generation. In hawkweeds, we see that the F1 is variable, but the F2 is constant. So it is completely opposite. Now, what was happening actually was in 1904, so far after Mendel's death, it was discovered that hawkweeds have apomixis. And so in 1904, Ostenfeld, a Danish researcher, he said that the different results that Mendel obtained in hawkweed, so different from pea, may be explained by the occurrence of parthenogenesis. And basically what it is what we call apomixis, which is reproduction through seeds, but in this case through clonal seeds, so not through sexual seeds, but through clonal seeds. And for that, you need two things to happen. First is that there's no female meiosis, so you don't have segregation and you don't have independent assortment, so Mendel's first and second laws. And then you also need to have parthenogenesis, which is that there's no fertilization, but the egg cell develops into an embryo without fertilization. This occurs, we now know, in some 250 different plant species. And so I mentioned already hawkweeds and dandelions, but also, for instance, blackberries and certain grasses also have this very peculiar reproduction system. Okay, now I'm going to talk about constant hybrids and the molecular characterization. As I mentioned before, I studied the genetics of apomixis, and in 2007, we wrote a review of the genetics of apomixis, and the title of that was The Mendelian Genetics of Apomixis in Plants. which is quite a contrast because Mendelian genetics is normally used for variable hybrids. So what we said here is that apomixis is a trait, but it's a reproductive trait. So it affects the reproduction in the next generation, but it's controlled by genes, and it's inherited in a Mendelian way. So it's controlled by dominant genes, genes that are dominant over sexual genes. And also we know when we review the literature, the model that fits the segregation ratios is that of that apomixis genes are always in a single dose. So they're always simplex genotypes. So big A, small a, small a, small a, small a in the case of a tetraploid. So how does it work? For instance, if you have a cross here, so you have one parent is sexual with a small a, and you got the apomix, you use that as a pollen donor, you can make a cross, and you get triploids which inherit all the dominant apomictic allele. And this is the interpretation of Mendel's results, so of the Haratian results. So you can see there's segregation for flower color, but they all are apomictic, so they all have the genotype big A, small a, small a. Now, this was quite a tour de force. Last year we published a publication where we identified the parthenogenesis gene of apomictic dandelion. And we used the gene from dandelion also to identify the gene in hawkweed, in Haratian. And what we discovered was that in this case we have a triploid and we have 3 different alleles. And the apomictic allele is dominant over the 2 sexual alleles. But what was very interesting was that in the controlling region, so in the promoter region of the apomictic allele, there was a transposon insertion. So that caused actually apomixis in Haratian. So this is a small repressor gene, and we see that the apomictic allele has a jumping gene or a transposon in the promoter, which controls its regulation. And then I think this was a nice occasion to refer to Barbara McClintock, who discovered jumping genes long time ago. So that was cytological work. And she got a Nobel Prize for that in 1983. But when she presented her research the first time in 1951 at a Cold Spring Harbor Symposium, she described the discovery of transposition. She reflecting and looking back to that. So when she had got the Nobel Prize, after that presentation, she said that I was startled when I found they didn't understand it, they didn't take it seriously, but it didn't bother me. I just knew that I was right. And I think that's the same mentality that probably Mendel also had. But fortunately, McClintock, she was recognized in time, so she got the prize before she passed away, and that didn't happen to Mendel. Okay, what's also interesting is that some of Mendel's hawkweeds still survived in Herbaria. So what happened is that Mendel sent his hybrids to Karl van Nageli in München, and he propagated them, and they were dried in herbaria. And even today, there's some 30 herbaria in Europe that have Mendel's collection. So in contrast to the peas, which are gone forever, the hawkweeds hybrids are still there in the herbaria. And we have a project together with the Museum of Grenoble and also with Wageningen University. So we try to isolate DNA from these herbarium specimens, and hopefully to detect that they have the apomixis gene, which we have discovered in Haratian indeed in them, which we expect. We'll see, but that's still ongoing. Then about future agricultural application. I want to talk about heterosis, which is something that was discovered in 1908 by George Schul. And what it is that if you make a cross between two inbred lines, then to the left you get a F1. The F1 has the highest productivity. So this is corn, this is maize. So you see what I call heterosis or hybrid figure. F1 gives a very high yield. If you continue breeding with the F1, then you get segregation, you get the F2 and you get the F3, and they have lower yields. So by crossing these two parents, you get seeds that are highly productive. And that was discovered by George Schul. And interestingly, George Schul in 1906, he also made a pilgrimage to Brno to see, to visit Mendel's monastery. So we have F1 hybrid figure, and when you go on, F2, F3, etc., you get a loss of yield. Now these F1 hybrid varieties, they have been very popular. So here you can see a plot from United States Department of Agriculture over the years, and you see the blue dots. This is corn, and this is corn harvest yield. You can see the blue dots is before they used the F1 hybrids, and after that they use different hybrid types, and you see an enormous increase in yield. So this is very popular. And you can see also this is started in maize, but in 1962 it was introduced in sugar beet, in vegetables, later in cotton, wheat, sunflower, rice, rye, oilseed rape, leek, alfalfa, barley. So it's very popular, highly used. But the problem is that every generation you have to produce these seeds again, because if you have selfing of the F1 hybrid seeds, then you get segregation. You can see to the left over there. If you would have apomixis, then the hybrid figure would have been fixed. So you don't have to make new hybrid seeds every generation again. And that seemed to be, yeah, something like a holy grail, but a holy grail in agriculture, you know, it didn't come close or near, but only 6 months ago, there was a paper which was called High Frequency Synthetic Apomixis in Hybrid Rice. And by Fernet et al., what they did is that they introduced synthetic apomixis. So first they avoided meiotic reduction, so meiosis and recombination. And then they introduced parthenogenesis. So the results of that. So here you see a sexual, to the left you see the F1 hybrid, and then you get the next generation. You can see that in yields, you get variation, sexual variation, but in their case, they had an apomictic progeny. You can see that it maintains the yield. So that's really, I think that is a breakthrough. What they did is that they used 3 recessive meiosis genes that were knocked out. Then they used the dominant parthenogenesis gene that was expressed in the egg cell. And what is very interesting, that they get a very high percentage of clonal seeds, more than 95%. But this seems to be quite close to market. However, one of the problems is that they worked with the recessive genes, the way it is at the moment. And you can think also of a strategy like poppy hawkweeds. So then you get breeding by dominant apomixis genes. So this is indicated in this cartoon. If you have a gene, so in the father which has apomixis, then you get a F1. Some of the plants will have apomixis, and the ones that have apomixis you can select, and they have high fertility, they have high yield. So, and you can also backcross them again with and use them as pollen donor. So it's continuous breeding. Basically, yeah, it's just copy hawkweeds. So we've been doing that in dandelions. You can see a picture of here. You can see to the you can see variation on the one axis and you can see constancy on the other axis. Now for us, this is quite difficult to see, but I just made a comparison because we've been told about facial appearance. So this is the analogy like it would be. So it's not that you have a single twin or something like that. So you can have different twins, but then you can have many, many different copies of it. And then, of course, the plant breeder selects the best. And the advantage is that in this case, you can, in one step, you can fixate any trait, whatever its genetic complexity, because you just like twins, monozygote twins. And another advantage is that you get already at a very early stage, you get a lot of seeds. So you get a high propagation rate, and you can do the early evaluations, which is very important in plant breeding. So then at the end of my talk, just to summarize, Mendel crossed hawkweeds to demonstrate the existence of constant hybrids and not to validate the P findings. Constant hybrids are apomixed with a clonal breeding system that is controlled by genes and that are paternally inherited in a Mendelian way. And finally, apomixes or Mendel's constant hybrids may revolutionize agriculture in the future. So I have to acknowledge people at KeyGene, many of my colleagues, many of my ex-colleagues, especially to mention the breeding support team in which I'm working, to mention Arjen van Tunen for his support. People that worked on cloning the PAR gene, many people from KeyGene, but we did it together with Wageningen University and with Plant and Food Research at Lincoln University in New Zealand, and also with Taki Seed in Japan. We work at the moment at DNA from Mendel's herbarium. So with the Museum of Brno in France, and also with Wageningen University. And I also want to thank my Mendel co-authors, especially Noel Ellis, because he's such a prolific co-author. Also Julie, Adriana Jessop, Mark Swain, and Franja Wijsing. And this is my last slide.
Oh. That was a very nice presentation, Peter. It's open for questions or remarks. Yeah, Hanneke. Oh, please use the microphone. Yeah.
So it was quite common that people in monasteries would also do this scientific work, because to me that's.
I think this is specific for the Augustinian order, and also the monastery altogether there was some 15 friars, so it was quite small. I always had this impression of monasteries like in medieval times. There were hundreds of monks and things like that, and they're only copying these books. But these people from there were a lot of a lot of teachers there. A lot of types of amnesia and things like that. So they are heavily involved in, especially in teaching. I think also in research. Yeah, not uncommon.
Okay.
Something sort of linking to the first talk, which was talking about inspiration of Mendel's work on peas. What do you think inspired him to work on hawkweeds? Was it innately or?
Yeah, maybe also due to Darwin and questions about speciation. Daniel mentioned that just a month before. Yeah, but that's at the end indeed. Yeah, so of the hawkweed. So the meeting the month before Mendel gave his presentation, there was this presentation on Darwin. Darwin's creation or how organic creation. And I think also Mendel was just interested when you read the paper, the letters to Naegeli. On the one hand, it's speciation and new constant hybrids, but he's also interested in whether he can explain it in terms more polygenic, polygenic models we call nowadays. So one of the reasons why he made so many crosses was that he thought he needed a lot because he need to put it all into different categories. So it was clearly much more complicated than the two colors that he had in P or the winkled and the round P. Yeah, but then he realized that, and then he gave up because he realized that it would be impossible to generate that many offspring to classify them. It seems that he was quite convinced that the underlying mechanism would be the same. But yeah, as I showed, he was really confused about the results. In the beginning, he was very happy that he got what he expected, but then he didn't expect to get multiple of these constants.
So Peter, what would you say to the breeding industry? Because they are preferably using talking in Mendelian terms, the variable hybrids, because the F1 it benefits from the hybrid vigor. On the one hand, it gives a very high quality feed, with more homogeneity, better yields. But on the other hand, you lose the biological protection connected to that variability. Whereas in your constant hybrids, you can fix that hybrid vigor. What would you advise? Maybe that's a better word. What would you advise the breeding industry to do? What is the corn hybrid breeding industry, seed industry?
Yeah, well, I would say it makes breeding much cheaper because you just have to generate a single clone. If you like that, you have take seeds from that and you test it at a very early stage instead of making two of these inbred lines and looking at binding ability. That's a lot of work. So that's one thing. Also, the seed production becomes much cheaper because you don't have to have separate male and female lineages. You don't need cytoplasmic menstruality and all these things. And of course, the downside is that you lose your F1 protection. But on the other hand, I would say just add extra quality or put extra minerals or fungicides and things like that, just to put added value to it. And then I think that the farmers will still grow these seeds and they will sell these.
Future of the breeding industry is ecomixes. If we can.
Yeah, I might be a little bit biased, but
you just got a prize for it, right?
No, I think so. Yeah.
Okay. Well, maybe the last question over there. Yeah.
I'm curious. So, did Mendel cross the triploid to a diploid? That's what I understood from your figures. So some of those triploids wouldn't have inherited the dominant apomictic gene. So there would have been a bunch of sterile. Did he notice this and did he reflect upon this?
I don't think he reported on that. He doesn't report on numbers in his letters, but he will have. Well, actually, so there's a second gene involved in apomixis. So you have to skip meiosis, and there will be recombination between them. So yeah, not 50% of the plants will be apomictic, but maybe 25 or something like that.
The very last question over there. Yeah. Please go ahead.
Yeah, just to comment on the last question. Hawkweeds are tetraploids, so you can easily cross them.
Yeah. Dandelions are triploid.
Okay, and with that, I would like very much to thank you. I have a few other very pleasant things to do. I can hand out some presents. And the first present I have is, of course, for our prize winner. It's on behalf of KeyGene, Peter. You very well deserve this. Kun je het uitpakken? Pak het maar uit. Pak het maar uit. So KeyGene is a very fastly operating company, as you might be aware. Yeah. Okay. This goes with it. Yeah, maybe. Yeah. Okay. Geef maar. I have some presents for the speakers. Yeah. Maybe can I have the speakers in front of the stage? Yeah. So we have a present for you. Thank you very much for your splendid presentation. For you the same. For you the same. For you the same. And with that, I think we close this program. I would like to mention the Crop Innovation and Business conference we are organizing as KeyGene together with Flemish Institute of Biotechnology and the German Center for Excellence in Plant Sciences on the 16th to the 18th of April. So if you're interested, please come and visit our conference. And with that, I think we can go to the drinks and the bites. Thank you for a very pleasant afternoon. And I wanted to check if we see smiles on the faces. I think so. So thank you for that as well. Yeah. Okay. Thank you. The drinks are on that side.